Okay, this is a very special webinar for AI for healthcare. And for folks who are familiar with our webinar, we typically invite academia or industry leaders to come and talk about their project, their cutting edge research they have done in AI for healthcare. If you're not familiar, you could check out our website. I'll put it in the chat and you can check out. We actually have quite a few upcoming speakers, and next week I have Sami Koyoju from Stanford, and week after from UPenn and Emory and Stanford again, and the Microsoft Research and so on. A lot of a good speaker coming up. But this week we have a special webinar. We're going to talk about some exciting work that we have done and clinical trial outcome benchmark. Let me first introduce panelists and speaker today. We have Andy or Chufan Gao and his PhD students from UIUC. Andy, you want to introduce yourself?
Yes, I'm currently a third-year PhD student under Professor Jimeng Sun, and my research focuses on structured medical data as well as clinical trial data. Yes.
Cool. Jatushan, you want to introduce yourself?
Sure, Dr. I'm Jatushan. I'm a second-year PhD student under Professor Jimeng Sun, and my research focuses on biosignals, clinical trials, and oncology care.
Great. And then Trisha, you want to introduce yourself?
Hi everyone, I'm Trisha Dash and I'm also a PhD student in Professor Sun's lab. I work on deep learning for clinical trials.
Cool.
Nice to meet you everyone.
Great. So what we will do today is we're going to talk about one specific paper and one recent paper in the benchmark that this team have created. It's about clinical trials outcome prediction benchmark. I think it's quite useful resources that many of you may find useful. And instead of just giving you a presentation, we're going to have a short overview presentation by Jatushan and Trisha, and followed with actually a demo and by Andy to show you how you can use this data set and how do you get access, and you can follow along as a quick tutorial. Then if we have time, we can do some question and answer at the end. Okay? Thank you. So with that, Jatushan, you want to share your screen and start the short presentation?
Yes. Sure, Dr. Yeah. Is my screen visible?
Yeah.
Okay, so the plan is Trisha will provide an overview of the project, and then I will go to the methods and the results, which is followed by Andy's tutorial. Yeah. So yeah, Trisha.
On automatically labeling clinical trial outcomes, a large-scale benchmark for drug development. And you can actually scan these QR codes to go visit our project page, paper, and the code. So let's start with the background. So drug discovery and development are extremely expensive processes, and clinical trial results are actually very vital for regulatory approval and patient care. However, large-scale, high-quality clinical trial outcome data remains limited, and which is actually hindering the development of predictive models. Also, clinical trial data grows dynamically and rapidly. And as you can see in the image on this right-hand side, that the number of completed clinical trials grows over the years. So, and these can be actually affected by external factors like COVID-19 or regulatory changes, etc. However, with this dynamic change, frequent label updates are also necessary, but manually labeling is impractical at scale. So that's why we have developed CTO, which is clinical trial outcome benchmark, which is a fully reproducible, regularly updated large-scale repository, which has around 125,000 drug and biologics trials. And this benchmark comprises of a comprehensive trial knowledge base, an automated labeling framework to label clinical trial outcomes based on aggregation of weak labeling functions available in public. And also we have manually curated around 11,000 trials. As you can see in this table here, our data set, which is publicly available, and it is the largest clinical trial outcome data set available. So I would hand it to Jaturshan.
So I will briefly go through our method, and okay, so the main motivator is that, okay, given a clinical trial, if you can predict its outcome at its initial stages itself, so you can reduce most of the costs related to that trial or make it more efficient in the drug development process. But in order to do that, like given the trials and the landscape and the dynamic landscape of the trials, it's harder to predict or even harder to do manually label those trials. So in order to address that, we come up with a framework so that we can build automated labeling framework functions such that we can label based on all the big signals which are available in the web and utilize that to train predictive models to enable better clinical trial outcome prediction. So to achieve that, like first to extract all kinds of information about the trials, we had to create a really good knowledge base for all the trials, which is available at the clinicaltrial.gov. So what we did first is like we extracted all the trials and we created like a trial norm, like multitude of information like such as PubMed abstracts, news, and related stock prices and other trial metrics. So as you know, like in the clinical trial, they have some trials which are kind of like linked to their relevant publications, and also like it's not fully updated as of because like most trials they are not actually linked to their abstract, but which we found in our further analysis that like there are like many trials which are not linked to their publications. So we did like about a search based abstract extraction to create like around like 125,000 abstract repository for all the trial, like for the trials which we could find permanent abstracts. And in terms of news, like we queried for around like 85,000 trials using a Google API, and we found around like 4.6 thousand trials which had their relevant news articles. And finally, like we did similar to see like the changes in stock prices, and we try to match that with certain trials to kind of identify some kind of weak signals in terms of the success of the trials. So after constructing the clinical trial knowledge base, we build a framework based on different kind of weak signals to identify whether the trial was successful or not. So the challenge here is that the clinicaltrial.gov not directly provides the information on whether the completed trial has been successful or not unless their results are reported. There are like some clear labels which is like very rare in their dataset. So in order to achieve that, like we utilize different weak labeling functions such that, okay, one intuition is that, okay, given the trial and its relevant publications, you can infer like based on the results reported there whether the trial was successful or not. Another weak signal could be that, okay, closer to that completed trials, like you can look at the relevant sponsors, like news articles which came out related to that trial success or not. And based on that, you can infer like, okay, whether that outcome was positive or negative. And we gather like different kinds of intuitions to like develop a weak labeling set of functions, which is then we aggregate all these weakly labeled functions to acquire better quality labels for all the completed trials. So to evaluate like whether our weak labels are accurate or does it have a really good agreement with human labels, we conducted an evaluation on an existing human labeled public dataset called TOP. So where we found like a really good agreement with a human label around like F1 score of like about 90.9 on for considering all phase trials and like about 0.7 to kappa. And also like further analysis on that also kind of like got us to know like, okay, even the human labels are not as gold standard because as a point when that data was labeled, the trial dynamic nature, like the new information becomes available as the time goes on, which is not considered at the time when the human labeled that. And due to that, the outcome also varied. So that kind of like impacted that, which also kind of like showed like having a human label at a specific time point for trials might not be a good feasible option. So And on top of that, we also kind of like manually curated set of gold standard label as considered as the most recent human annotated label to conduct our experiments, where we initially had a rule-based labeling process based on the status and the primary p-values. And then finally, for the harder trials, we went through all the resources we collected through our trial knowledge base in order to infer whether the trial outcome was positive or negative. So moving on to our experiments, like, okay, so we wanted to see like, okay, which of the labels are better? Are the outdated labels, which is from a human from like some time back, or are the weekly label function, which is like, it can be frequently updated and which can be like improved, like depending on which is more scalable, or is it like more recent human label data? So we had the human label data, which is from the recent information as like upper bound of performance. So, and we train different machine learning baselines on each and every data set. So here, the blue points refers to the machine learning benchmarks, baselines trained on the CTO, which is our weak labels. The red refers to the baselines trained on the top benchmark, and the gold refers to the baselines trained on the new manually annotated data set. So as you can see, okay, so how the process works is that, okay, so we take all the data from before the trials completed prior to 2020 we train on that, and then we evaluate on the evaluation set for the trials which are completed on 2021. Similarly, like we do it in a progressive manner, like where we did it for before 2022 and evaluate it on year 2022. So one of the good observations you can get from these figures is that compared to the recently updated manual data, like ours, it's like more competitive, like it's more competitive like in terms of like phase 2 and phase 3 compared to like a all outdated human annotated data. So which is kind of like shows the importance of like having a frequently updated labels for clinical trial to in order to build better predictive models. And finally, we also conducted an experiment on different conditions, like considering like top 20 conditions from the clinical trial data, and where the goal line specifically shows like the possible upper bound of performance we could achieve, and the blue shows the achievements of the baselines which is trained on our weak labels. And finally, the red kind of like shows like performance on the top benchmark, which is the older human annotated data. So this also kind of like validates our claim. Having a more updated labels kind of improves the performance of the predictive models. So now I will hand over to Andy.
Patricia, if you can stay on the slides for a moment, there's a few important points. Maybe we can have a quick discussion the last 2 slides on the result side.
Yeah.
If you go 1 slide back, there's a lot of information here. Can you say a little bit more about I guess there's several questions I want to ask. One is this yellow curve and all those yellow symbols, this represent the model performance trained on the manually curated labels, right? Versus the blue points are your automatically labeled training set. And what's the problem of just using those manually labeled data points? And why do we need CTO, or why do we need this automatic labeling process?
So it's basically the manual effort, like as Trisha initially mentioned, like, so you can see that the number of trials each and every completed, it increased like for 2024 itself, like there are like 14,000 trials, and like it's manually infeasible to obtain, label all those trials. So we are, CTO, like we can automatically label them in within hours. So that's like the advantage of like having that. So because, and also eventually like if we do labels like manual, we won't be going back to the same trials again to do the labeling again. But like in CTO, like you can frequently update the labels. Yeah. So those are like the advantage we have here. Yeah.
Yeah, I think there's an audience asking question. Misha asked a related question. But can you explain, maybe you presented an earlier slide, if someone wants to manually label the outcome for a trial, what exactly do they need to do? And what background do they really need to have? Can you show, I think you have some slides showing the labeling process, maybe, yeah.
So yeah, so we can have some certain, like, in order to make the manual process easier, like we could have some clear indicators from the data, which is like the status. Like if you take, like, if the trial is terminated, like it's most likely to be a failure, and if it was suspended or withdrawn. And it gets more complicated when you try to label like completed trials to assess whether they are successful or not. So one thing would be the p-value, and also like for depending on the phase, depends on like looking at the adverse events. And then it gets more complicated if there are like no actual results reported. So it's more of like going through their publications and seeing what they reported there, whether it matches the primary outcomes of the actual trial and whether they have achieved it or not. So that's like the process of how we did the manual curation of the data.
So I want to bring in a few more points. So like obviously manual annotation takes a really long time. So if you just take a person that's probably not a clinical trial expert or like just a general like consultant, asking them to label one trial with all the due diligence that they would have to do, like climbing through, running through the news and the stocks and the press releases of the company, that would probably take like the better half of a day, right? That's very not feasible to update, which is why we try to speed up this menu curation process up a little bit by having these first initial rules. And we also verify these, like the status and the primary p-value based labeling with some of the industry. One of the first slides, there was like some industry private data sets. We were able to get a subset of those, and we validated the status feature where we take like the simple like terminated equals failure labeling function that had like a very high percentage of accuracy, as well as the p-value. Yes.
Yeah, it doesn't definitely is a very complex, involved process. Probably require a lot of skill set, right? Domain knowledge. Mishala, you wanna? Yeah. Thanks for the good question.
I would love to ask a question. Thank you. Come to a couple of your lab talks, so completely an outsider, but just for background, I'm a physician by training. So my question here is, when you say manual annotation, and you have data points of like 2022, 3, 4, like so it sounds like, I mean, I just want to get an understanding of what you mean by that curve. And I think one of you explained just now that it is about collating all the data sources, right? So for example, I would imagine, yes, it totally makes sense that it would take about half a day conservatively to annotate. But what do you mean by the changes between, so is it like in 20, let's imagine all the information sources that became available in 2022, and then the human was asked to annotate again, and then 2023, and then 2024. Is that what you mean? And did you do any labeling for this, or was this part of some data set? I mean, I'm just trying to get a sense of
Yeah, it's a really great question. So the yellow line that you see here, that is our manually curated data set. So we have two contributions here. The blue line is our automatically gathered labels. So remember how Jathershan talked about how we had like these, weekly supervised labeling functions? That's like all of those labeling functions, like the GPT, the news, that is automatically aggregated. And that's like that predictions is in the blue. And from those predictions, we also manually annotated 11,000. That would be our new gold standard in the yellow here. And we didn't go year by year. Well, we didn't re-annotate them year by year. We essentially just took the last 4 years from 2021 to 2024, and we just annotated them. We sat down, we looked at all the knowledge sources that were involved in our knowledge base, and we looked at we literally just Googled each of the clinical trials to see whether it had relevant publications and press releases, and that's how we manually annotated the yellow and gold.
I also want to add one point here. Maybe the curve, when you plot this over time, is a little bit confusing. So here, the y-axis is the predictive model performance tested for the trials that completed in or on that year, right? So we're basically predicting the trial outcome based on the previous years of labeled data. So that's why there's up and down performance changes over time. But the labels are acquired over time for the trials that completed in that year. And we are not going back, check old trials and label them again. But every year we just label that year's completed trials. So that's where we gather more and more training data.
Yeah,
Yes.
no, that makes total sense. And I would definitely, that's what I suspected. It's just the connecting line between them. I wanted to make sure, because that's an advantage of, say, an automated system, right? You could go and continuously update, versus you could really, nobody is going to go read the additional data that now became available and update the rating or change their rating. So
Yes.
yeah, you should probably figure something out to like make that point clearer. But
Yeah.
no, thank you. And who did the annotation? Did like basically all of you?
Yeah.
Yes,
basically.
we all did, and
Oh,
we
okay.
also, we consulted with the clinical trial expert. That's one of our authors actually in the paper.
Lovely.
And one additional point here on this curve, since we're here, is this bottom, the red line. That's a fixed data set that manually annotated by experts as well, but for what, up to 2014, 15?
Yes, up to 2015.
Yeah, so those are relatively older trials, and so you can see even their human expert labeled data, because they are kind of outdated, right? Outdated by 6-7 years. Then the performance of the model trained on those old data actually is perform much worse. So you really need to update to bring the newly completed trials into your data set, right, in order to create a better predictive model. So that's why this automatic annotation labeling is very essential.
And just to add one point on the graph, like, so the dotted line which connects is just kind of like provides like the mean performance for all the baseline. So yeah, for train on that particular data sets. Yeah.
Okay, great. I think this is good discussion. Shall we move on to Andy's demo before we?
Yes,
More
let's
discussion.
do that. Yeah, that was a really good question. And if you guys want to play around actually with how we created some of these plots, that's totally available. So if we go to our website, which is linked in the meeting chat, and you go to tutorial notebooks here, you can see in the README, there are 2 notebooks that we highlighted specifically just to get started, playing around with our CTO framework as well as our manual labels. And we're going to be going over both of these notebooks today, and they're going to have a lot of overlap, so it's not going to be too much. Yeah. So let's just quickly go over just the getting started CTO versus top. As you recall from Jitharshan's wonderful presentation, top is just the previous largest publicly available expert annotated clinical trial outcome data set, and ours is obviously the one we want to compare against it, right? So in this notebook, we're just going to compare training on our automatically labeled trial data compared to TOPS manually curated trial data. And to make things fair, we're going to be evaluating both models on the TOPS gold standard test set, just to make things completely fair. And I'm just going to quickly run this. This shouldn't take too long. If I just restart and just run all, this should be able to just run the entire notebook in a few minutes. But I can just go over quickly what each cell does in the meantime. So obviously first you gotta load everything in. We git clone our repository, the baseline top repository, as well as our clinical trial, as well as the clinical trial statistics that we downloaded from clinicaltrials.gov. Yes. If you want to use the latest version of clinicaltrials.gov, you can just you can uncomment this cell and just run this cell instead, and this will point you to the new path. This essentially just goes to the website and just manipulate this version. In this cell, this is just loading our final, automatically aggregated labels for each phase. So this is our final phase 1 predictions, our final phase 2 predictions, our final phase 3 predictions. This is our best predictions on each phase as compared to the other type of label aggregation methods. We found that random forest was the best. And to obtain a set of features that we're going to predict over, remember, we want to compare these binary labels, right? So the text features that we're going to be predicting over will just be, the very essential clinical trial aspects, right? We need the specifics of the trial. For example, what phase the trial is in, what diseases the trial focuses on, what the intervention was. For example, well, these interventions are like aspirin or other things like dinesipil, stuff like that. Just different drug names that are cleaned in lowercase. And also, if there's multiple interventions, we just concatenate them. And finally, we have the eligibility criteria, like what patients the trial is actually focusing on. Is it focusing on, older patients? Is it focusing on, women versus male, like, etc. What the eligibility criteria is. And finally, the study architecture, right? What's the study design? Like, is it double-blinded, single-blinded, etc., stuff like that. So that's exactly what we do. We load in all the CSVs related to clinicaltrial.gov for the diseases, interventions, criteria, design. And we pre-process all of them. Like, for example, we only use the down case mesh term for consistency. We don't want any, capitalization errors. We join all the interventions. We drop nans for cases where the criteria isn't there. And we join everything into a single text feature. That's just called, feature, where we just concatenate everything in a single line. And what that looks like is, if I just run this cell a couple times, it's just features, right? Like, this is one of the features in the trial. We can go iterate through more of them. For example, like, this is a phase 1, phase 2 trial. Yeah. It's just a bunch of text features that we will be featurizing using TF-IDF later. But first, we also provide a bunch of visualization tools where we can actually see how the trials are distributed.
So Andy,
yeah,
just clarify one thing on the feature construction there? I guess you're just creating a very simple feature representation that include all this different trial related information as a text string. And presumably someone want to build a more sophisticated trial outcome prediction model can replace this part, right? Maybe introduce a whole bunch of a structured feature, right? Phase and drugs specific information and so on. So they can further improve your trial outcome predictive model pipeline, right? Instead of using the text string, they have more sophisticated feature construction, but they can still benefit from the labels you created. Is that how they should think of?
Yes, absolutely. That's a very, very excellent point. For example, if you want to use a large language model on the criteria or the trial design criterion, that can be totally doable. But in this case, for just the sake of the notebook, we decided to keep things simple. But this is totally doable. This is just feature construction, and the labels is completely separate from this. Yes. Yeah, I can just, here's the phase, here's the completion year. We get to see the same plot that there's plotted, so we can reproduce all the plots we have in the paper, which is quite nice. And here we just have a visualization of like the top 30 most popular mesh conditions that we find in the trials. And surprisingly, like, I didn't know this before, but cancer is just by far the most popular type of clinical trial. It takes up 3 of the most relevant mesh conditions is cancer related. I think it's very cool. But yeah, here we just use TF-IDF, but please feel free to improve on our methodology and add whatever advanced modeling function you would like. But in this case, okay, so now we get to the data splitting. And in the data splitting, we always test on the supervised top labels as before to keep things fair. And we also just have like a simple switch where do you want to train on top or do you want to train on CTO? If you want to train on top, we just get those NCT IDs and then save it for trained DF. And for CTO, we do the same thing, except for one step. We make sure that we only select trials that have the completion date before 2015 to keep things in the same distribution as the top paper, because they only use training trials before 2015. Right, and here we just merge our training NCT IDs with the features that we obtained previously. And we use TF-IDF with 2048 possible features with after processing of the English stop words, of course. And after this is just a very classic machine learning binary classification outcome, right? We have like the F1 score, we have the average precision ROC AUC. We can choose from random forest, we can choose from anything really, but I just decided to use the sklearn default classifiers as a simple baseline. And we also do it by by phase, right, to plot what the phase specific performance is. And from here, we see that our CTO results is here. And if we run this again with the top option, we can also get the top results here. But I just saved it just for convenience here. And from this, we get to see some kind of cool trends. So even though our CTO labels were not manually annotated on a top dataset at all, it was only guided by the top training dataset, we're able to get actually stronger performance on some of the phases. For example, for phase 1, we get to see an F1 score of 0.7 versus an F1 score of a 0.6 in the original top dataset. So actually, surprisingly, we were able to do slightly stronger than top in some of the phases. Actually, for F1 score, we beat top on every phase. Yeah, this is a very good sanity check because we didn't specifically ask it to be similar to top training, but it just turned out to be. So this just completely validates our framework.
So just on the top data set and it's coming from previous papers, right? I put it in the chat and it has a methodology, more sophisticated methodology for trial outcome prediction, but it also created a smaller, much smaller benchmark data set, right? For evaluating the trial outcome prediction. And they use the data up to 15 or for that?
2015, yes.
Here, you improve the performance on top test set using the similar strategy to gather the training data. The performance gain is significant. Do you think it's mainly because the CTO, the newer one, has more trials, or it's because the quality of the labels, or is it like much larger training set than top, or?
Yes, I think both of it is kind of relevant because the top training set by itself is quite small. I think it was like only 12,000, and in our case we have 125,000, right? But even if we take the samples that are before 2015, that is still much higher than the original top training data set. I can run those numbers for you in a bit, but I can tell like that's much higher. And in terms of the labels, right? I believe that the labels are probably a little bit more updated because in the original, just like even in the original top labels, it's not completely perfect. There are some examples where we go back and look at the top labels and some terminated trials are marked as success. So it's possible that the trial, some parts of the trial change later on.
And just adding on to that, so even though our data set large, so one advantage is that we cover lots of different trial conditions and different diseases, which is not covered much by the top, which also might have contributed to the performance increase as well in terms of performance.
Okay, cool.
So the second notebook I'd like to present is our 4 experiments notebook, where we try to reproduce all of the things that we see in our paper, as well as like the nice like line plot that we saw in the earlier presentation. Essentially, most of this is exactly the same. We load in the data like usual. We build the text feature like before exactly. And just now we have to load in the manual labels and make sure there's no overlap between the manual labels as well as the CTO labels, just to ensure a good concrete data split. And in this case, because we want these labels to be as accurate as possible, we also do a progressive label processing on our CTO labels. Because yes, the CTO labels are good, but we can make them slightly better, right? Some of the obvious things that we can do, like we did in our manual labels, is we can just mark trials that are marked as, terminated, withdrawn, suspended, withheld, or etc. We can just mark those as, 0 for not a success. And if the overall status is this very, very rare status of approved for marketing, then that is quite clearly a success. So we can mark that as 1. And similarly, we can use the similar strategy with the p-values. So if there is a primary outcome p-value, like what the trial is investigating, that is less than 0.05. Now, the 0.05 is a broad criteria. It's not like universally adapted because some trials would have like more stringent p-values. But in this And using this quite simple rule of less than 0.05, we found that empirically this does have a pretty good agreement with private trial labels as well. So we can use this to kind of as like a simple like rule-based threshold here. So in our case, to further process some of our CTO automated labels, we can apply our status condition where if the status predicts a 0 or a 1, we can just set that as the new label. Similarly, if the p-value predicts a 1, we can also set that as the official label. And finally, for every other label that is not covered by the status or the p-value, which is most of them, we can now use the CTO predicted probability of the trial success. Yeah. And then we save this for later to load in for our downstream data process here. Like for the manual labels, we obtain all the NCT IDs that are in the manual labels and correspond them with the features and save that for later as well. And similarly for top, we do the same thing. We load in all the NCT IDs from top and concatenate that with the features that we've processed previously. And now running the baselines, we can just show you like this is exactly the results that we ran for the results in the paper. So for each year and for each domain, we train on the data sets that end before that year. So if we have the year 2022, we train on all trials that completed before 2022. And we test on trials that completed within that year. So this is only possible with our manual labels and our automated labels because the top labels do not have trials that are complete after 2015 in their training set. And if we run this code, which takes a while because it is slightly more involved and there's like 4 years and 3 different types of labels, we get a very nice data frame corresponding to the train mode and the F1 and the F1 standard deviation as well as like the precision and average precision and the RLC score. But this is kind of hard to see. So if we run this function for plotting, we see something that corresponds very strongly to the results we saw in the paper as well as the slides. And this is the thing that we're most excited about because we see here again like the top model does not basically change at all as we go from year to year, whereas our manual labels and our automated labels are able to keep up much better with like the year to year changes. And this is one of the most exciting parts. Yes. Okay, so please feel free to try these tutorials by yourself. These should be just completely runnable. The first notebook should take around like 2-3 minutes to run, and this one should probably take around like 12 minutes to run. You can feel free to change the features that we use, the models that we use, as well as like add your own implementations of any of the feature processing. But yeah, these should be fully runnable. And thank you for listening to our talk.
Thanks, Andy, for the nice demo. And maybe, so for the audience, if you have questions, feel free to put in the chat or just ask directly. And I can maybe start off by asking some general question, and all of you can maybe answer that. So this is more of a motivation of this work, right? Why did you decide to build such a public data set for clinical trial outcome? What are the unmet needs that you're aiming to address with this data set?
Yeah, that's a really good question. And Jitharshan, if you can share the slide of the list of like publicly available versus privately available clinical trial outcome data sets.
Yeah, sure. Just give me.
Yeah, so this slide, right? When I first started my PhD, I remember, Professor, you were asking me to do some, clinical trial outcome prediction. And all we had was the top data set from former PhD student and IQVIA. And we only had this data set, right? This was basically only for small molecule drugs. It only had like 12,000 trials that we could actually use. It says 17,000 here, but I think after some processing, it came down to 12,000. And that was just not enough. When we tried extending it to like more advanced drugs, for example, when we tried extending it to biologics instead of drugs, we just found that there was only like a few hundred to maybe 1,000 trials that were actually labeled. And we tried scouring the internet for more like publicly available trials, but all we could find was just paid options. For example, like Biomed Tracker and Silico Medicine, yes, Trial Trove, these do have a lot of trials, but we just weren't able to access them because we didn't have the subscription. So we were essentially forced out of necessity to make this data set, and we hope that this can help alleviate like future PhD research efforts, or as well as like industry efforts if they want like open source way to like automatically label trials. I think that's a very good like floor for things to be on, right? Because the top, as we see, like the top, if it's not continuously updated, the trials become relatively like less and less valuable later on. So having that floor of the automatically updated trials is very, very useful.
Trishan, you guys have anything to add to this motivation questions?
So Andy pretty much covered it. So I was at my initial stages of my PhD, so starting off, and it was more of an exploration, and Andy pitched us this idea, and like then I started more exploring, and it got more interesting, like trying to find the actual label for the trial, whether it was successful or not. That was even like a hideous task at the initial stages. So which further motivated us to kind of like build an automated way, like to have a good foundation, so that from that way we can further improve like the automated labeling framework as well, like have better weekly labeling function, have better resources, it will be like a upwards trajectory in having better labels. Yeah.
Trisha, anything you want to add?
I think it's like Andy actually covered most of like all of it, but I think like when we tried to manually annotate some of the trials, we find that it's actually absolutely necessary to have like automatic labeling option, because like it's really hard and really time consuming. So like when we actually tried doing manual annotation, it was like it's absolutely necessity actually. Yeah.
Yeah, very good point. I think you probably have a first-hand experience, right? You guys labeled what, 11,000 or how many? 11,000 over a couple of weeks. Definitely.
Yeah.
A lot of, I would say you guys are quite efficient just to get those done, but still it's definitely not feasible if you want to do it in real time and quickly, right, for all trials and over time. Maybe yeah, go ahead.
I would say like the 11,000 trials was only made possible because we had collected all the labeling functions, all the news, all the publications as well. I think that made it
right. If you have to gather the raw data and like put them together, that's probably not just 2 weeks of time, right? Probably years, and to do that. And, okay, so I saw Patrick has a question there. Let me see. What's his question?
How long the manual? A couple of weeks. I think we had started talking about labeling the trials for a while before we actually started doing it. Of course, like in the beginning, the progress was quite slow because we weren't sure like how many to label. But I think eventually, like I think closer to like our internal deadline, we like cranked it. I remember like this was actually during time when I had to like just sit down and just look at the trials for all like. I looked at like 100 trials a day, like just manually checking. Like I did this for probably like 12 hours that day, and that took a long time.
Yeah. And yeah, as Andy previously mentioned, so like since we had the resources already collected at Meditisa, so even during the process we wrote some scripts to kind of like make the visualization easier, to retract all the resources fast, so that we can look at the information faster and make the labeling faster. So yeah.
Yeah, maybe you guys can, this is a good segue to the engineering, the data set construction piece. Can you give me some example, maybe Trishan or Trisha, because each of you handle some specific data sources, right? And to create those weak labeling function for that data sources is kind of a lot of effort, and engineering and creativity has to be done. Can you give me some example of some of those labeling function, and how did you create that labels, and how do you implement that?
Yeah, sure. So I can start off with like the LLM-based interpretation on PubMed, like as one of the labeling function, and the trial linkage, which I think the most creative one, because what
Do
we
you want to
put in
show
okay.
that? Sorry for the
Yes, sure.
yeah. Should show that the overall this right so people understand the this one or this one yeah.
Okay. So yeah, so pretty much like sort of from the PubMed abstract. So one of the engineering problem which we found is the clinical trial. Okay, they have data like which kind of like combines each and every like which matches each and every publications abstracts with the trial, but like later we found like there are lots of new trials which are not, it's not updated, and we have to manually search. Even for some of the trial it has missed lots of information, so we had to do like a manual search, not manual search, like we kind of like automated the process of search and extract more PubMed abstract. And then after it was relatively straightforward to like prompt an LLM to kind of like provide us feedback like on prediction on like the outcome of that. So that's one of like the big label. And then another one which I think which is one of the creative thing which it is okay from a person like okay, if a trial phase 1 trial has progressed to the phase 2 trials, that indicates that the phase 1 was successful. But the challenge is that like there's no clear indication of like a deed or like a identifier or anything in the data which shows like okay, this particular drug has progressed to this, or like this particular drug has undergone these particular trials which are like it's phase 1, phase 2, phase 3. So we had to kind of like find out a way to kind of like automatically link these trials and also even match it with the FDA approvals, which is also hard because there's no direct match with the NCD IDs of the trials and the drug IDs. So we came up with some creative ways to kind of like match. It's not the best optimal way to kind of like highly accurate one, but it's kind of like provides like a weak signals on to show the trial progression throughout different phases and everything, which I think one of the creative labeling functions we came up with. Yeah.
Yeah, what maybe, or I don't know who did that part. I remember you guys even have to figure out some standard way to, or standardize the drug names, right? Because when they go through trial phases, the drug brand name, generic name, all those things, or maybe that's compound, some kind of a specific code name has to be linked, right? And can you say a little bit more on even just on that specific aspect so you can know they're talking about the same drug?
Yeah. I think I used Drug Bank. I had to mine. So Drug Bank allows very generously a free download of their like some of their like research reviews, and I was able to do that and just mine literally every single possible like brand name that's like known of like a certain like every single drug, all their brand names, all their international names, all their names, and then create like a massive dictionary that I sent over to Jethro.
Yeah, so pretty much the issue we had it when it was doing the trial linking, like different trials, the terms like they use for the drugs and everything, it was not like standardized. So that's where we had the troubles with, yeah.
And yes,
I think the compound name is generally matched to the drug bank will have like synonyms for each like single drug that should be in there.
Thank you. Yeah, that can be. And can you give me an example of what the label and this, I might not be getting this right. Let's assume that a trial went on to phase 1 to 2 to 3, but then you didn't have any information on the approval. So what were the like how did this labeling how did this labeling process sort of like change? What degree of sort of like, yeah, do you get my question? It's like I'm just trying to get a sense of what granularity did you end up measuring out of this process. It's super interesting. So
yeah, I think Detergent did it a really smart way, right? So if we go to all the phase 2, if we do it by like, okay, for all phase 3 trials, we look at phase 2 trials. If we can find a phase 2 trial that matches the phase 3, like that matches the intervention, that matches the like eligibility criteria, that matches the sponsor, something like that. Detergent can go more in depth, but that phase 2 trial is now marked as marked as linked to the phase 3 because we know that the phase 3 exists and we know that the phase 2 exists. And yeah. How'd you do it for the FDA Orange Book again?
Yeah, so the trouble comes like when it comes to the phase 3, like So we have to be like because like it's clear not indicated like mark. So we were only focused on identifying the positive trials when it comes to the phase 3 only. Either if it has like a matched phase 4 trial, or based on the FDA approved, like extracting some drug information and generic name, some features out of it and try to match it with the closest trial based on the approval date and the trial completion. So those are like some tactics that we try to identify some kind of weak signals there are to match it with the FDA approved. Yeah.
Okay, thank you.
What about the,
So-
I guess maybe Vishal is implicitly asking also the negative. For phase 3, how do we get the negative outcome if there's no FDA?
Yeah,
approval?
that was exactly my question,
Yeah.
Jimeng. Yeah, exactly. I just figured that because I think that is the harder part.
Mhm. I think we had some heuristic, right, Jatusha, like over some gap of a couple of years. If you don't have that label, then we consider it's probably negative or something. Did you, or we just
Yes.
don't have a negative for phase 3 or no?
We had it, but like the only one challenge is like so for the phase 3 we kept it as okay. So in our aggregation format, like we had a label something like -1, which indicates like there's no clear details on whether which this trial was successful or not, which is like which was used for phase 3 only. Because it's kind of like a tricky question where even though if there's no phase 4, even though like there's more possible like it could be a successful. So that's what we did only for the phase 3 ones. For the other ones, like we kind of had it like like a label straight away, like based on whether there was a matched one or not. But for the phase 3, we had it more of like an inconclusive label. Yeah, it's a bit tricky in terms of the phase 3 task. Yeah.
Because even for phase 1 and 2, we should have some kind of waiting period, right? They may be successful, but the next phase hasn't started, so we probably can, if we haven't already, some gap, right? To say, oh, we'll wait for a year and a half, right, to see if there's a follow-up trial. If no, then maybe consider negative.
Yes, yeah. For the initial trials, like so, we have something like to kind of like find the next linkage. We had like a time-based filtering, which is basically like to find a trial like if they has some relation like for next 5 years, which we had it as a bigger or larger to see whether the trial has progress or not in our big label function.
Cool. I noticed the time is kind of running out of time, but I'll just ask maybe a final question. But first, thanks for all the speakers and panelists give this in-depth analysis of this nice data set. What was the most surprising thing that you discovered when you work on this project? Can each of you share some your own experience?
Yeah, I can go first. I worked a lot on the news. There was a couple of surprising things about the clinical trial news. One is obviously scraping news websites is a huge pain, and second is that I was really surprised that there isn't like an already existing aggregation of clinical trial news. I assume because news data sets are so prevalent, I assume there would be some sort of like repository that like had it like already scraped or but that was just my incorrect assumption. We had to manually spend a couple $100, I think, to do that ourselves.
But also, I think one other thing on the news part, you didn't find too many news, right? As comparing to the number of trials that the ones have, I guess, newsworthy is much small portion, I guess, right? Which maybe makes sense. Only maybe the major phase 3 trials has some. Is that the case? Or those 4,000 or 600 newses? Those are mostly on the phase 3.
Yes, I will say that I think most news I find is positive. So if your drug had a negative result and you're not a large company, then reporting that on the news is probably not
worse
viable, like helpful for you. But like sometimes I do find negative news from like large companies saying like, oh, like Moderna had a trial that like stopped due to some reason. But I've never seen like a small company like saying like this drug stopped, and it's like on Google News or something. Yeah.
Okay, probably not a newsworthy, right? Probably a lot of trials failed, right? So it's okay. What about Trisha? Your anything surprising in the process that when you?
I think like the first surprising thing was like the clinical trial outcome labels are not publicly available. That was the
That's true.
first surprising thing. Yeah, I worked on like the clinical trial amendment counts and on the stock prices. I think like it is a like it's there are no straightforward way to get the like company names to their like tickers. I don't know why but like there is a like the other way there is like from ticker to the company names. Yeah, and other than that, I think I used Yahoo Finance and which did like where we can actually got 20K trials. I think other than that, some smaller companies, like some big companies sometimes buy smaller companies. So when you look for the ticker, you like that is not available, but at that time it was available. So like there are some issues with that, like some
That's
classes.
true, right? There's a lot of acquisition maybe happening in this space, right? We have to figure it out the over time how it happened. Interesting.
Yeah.
Jatusa, anything you find surprising or?
So my first surprise was like the clinical data, which we've got, like which is started off initially. It's like not super clean, and we had to do lots of processing, like in every aspect. So that was the bigger challenge. And then when I found like when you in the process of when you found like the FDA Orange Book, and then another surprise which came up is like, okay, the IDs which are used they don't provide any information on what kind of clinical trials. It's not matched directly with any NCT IDs. So that was like the second trial. If there was like a good mechanism there, that might be the best straightforward way to get a trial outcome labels, but like which was not available to kind of like match that. So which was the second surprise which I had. And in terms of the method, like what in methodological way, what surprised me, like, okay, without having like a really hard like labeling with humans, like to do find the labels, like if we can still use some weak signals and create like really good quality labels which can like further enhance the predictive modeling. So that was like through the method development what I found as a surprise.
Yeah, very, very good point. I mean, maybe we should contact FDA somehow or to figure out what's happening there. Well, maybe there are some secret or not very well-known sources that has all that information, right? So, but thanks, guys. I think I find the most exciting part of this work is you guys created a lot of a novel and creative way to correlate the trial outcome to some kind of a proxy, right? Whether it's a news, stock price, LLM output, phase transition, right? All these things are kind of interesting, right? You treat them as a independent, smaller task, then aggregate them together, and which make total sense. Thank you. I think that's the end of the day's webinar. Thanks everyone for attending the webinar, and thanks Andy, Trisha, and Jatushan for the wonderful talk. Thank you everyone. Have a good week. Bye-bye.
Thank you.
Thank you.